please help me welcome Professor Tamara Galloway from the University of Exeter. And Tamara is a professor of ecotoxicology, and she will talk to us about microplastics in the ocean. This is a topic that hasn't received much attention for many years and was actually one of these topics that people disregarded and said it's not worth funding, that it won't make any difference in the oceans. But today we know that it's a huge problem, and there was a science paper just last week on increasing trends of plastic pollution in the oceans. So it's an important issue that we will see much more in the coming years. I also want to, before Tamara comes up here, Tamara's biography is in the ASLO pamphlet, but I also want to urge those students and scientists that are thinking about taking career breaks to raise family to look at Tamara's example. She took 10 years off to raise her kids and came back to pursue her passion, and with much success, and hopefully will continue in the future. So Tamara, please.
Thank you very much, and I'd like to start by thanking the organisers for inviting me to talk to you today. It's a real honour to be here. So if I move on to my first slide, I've come here from the beautiful southwest coast of England, from Exeter, and as you can see, we're very lucky to be surrounded by beautiful golden beaches. It's very tempting to go for a walk on these beaches, but what you can see from this image, and this mirrors many of the beaches around the world that we care to see, what you will frequently see is that while you're walking across the beach, you'll come across marine litter. You'll come across vast islands of floating plastic debris and other waste that's present on the beach. And together with some colleagues at the University of Exeter, we decided to determine, well, how much is too much? How much plastic actually is there on a typical beach? And this is a picture, colleagues with, including Matt Witt at the University of Exeter, got a group of volunteers. We looked at a 25-metre stretch of a single beach in Cornwall, took 3 hours on one afternoon. Took all of the pieces back to the laboratory and identified all of those pieces that were plastic. And the results you might find are quite astounding, because the actual number of plastic pieces from that one beach on that one afternoon was almost 577,000. And that included items including a toilet seat, down to cotton buds, down to plastic bags. So you're starting to get a feel for the scale of the problem. That's one beach, one afternoon. And it's not just our coastal oceans which are suffering, because many of you will have heard of reports of the Great Pacific Garbage Patch, a large area floating in the oceans, roughly estimated to be around the size of Texas. And what's happened is that floating debris, 50% of the plastic we throw into the ocean, has a lower density than seawater. It's pulled into the downwelling zones of the ocean gyres, and it floats there. And it's been estimated that some of that plastic takes about 6 years to make its way there from the coast of America. The currents make it a much quicker process from the coast of Japan. It takes about a year. So floating islands the size of Texas. How did we get to such a state where we were able to casually disregard or throw away all of these items into the oceans and make such an impact? Well, we can trace that back to around about the 1950s, when advances in material science enabled us to start mass producing plastic items. And almost at the same point that we realized that we could do this, we decided as a society that it was going to be absolutely okay to throw those items away. We created the throwaway society, to such an extent that not only was it acceptable to throw things away, it almost became expected. We were synthesizing and making these beautiful items which could last for up to 500 years, designing them to be chucked away after a single use. And we can track forward 60 years from the 1950s to our current day, and if we look at the recycling rates across Europe only, we discover that we're throwing away 30 billion plastic bottles to landfill every year in Europe alone. And I think some of those figures start to have a credence with the previous talk on organic carbon when we're looking at millions of tons being discarded into the sea every year. We make around about 300 million tons of plastic every year, derived largely from oil. And what happens to that plastic as we throw it away? It gets washed out of landfill, it gets washed into waterways, and it gets washed out into the sea. And we know that this is happening thanks to some of the beach surveys and the scientific surveys that have been being conducted by organisations such as OSPAR. What you can see here from 2007 are some figures for the numbers or the types of different beach debris that were collected around oceans across the globe. And what you can see here is that the predominant item across all beaches are plastic and polystyrene pieces. And it's not just the beaches, as I said, but if we go down to the depths of the ocean, we find that sediment debris is also predominantly containing plastic as well. So you can see from this benthic litter trawl in the Mediterranean, the predominant item here is plastic, and it comprises of different kinds of packaging, food packaging and beverage packaging. So these are things that we've dropped, we've thrown into the oceans, and they're there in very large quantities. But just because it's there, it doesn't necessarily mean that it's causing any harm. When we start looking at what kind of harm plastic does, it's quite easy to determine what harm plastic does when it's a large item, when we can see it. So we can look at some of these very emotional images here. This is a picture of a fulmar. Petrels are birds which like to feed from the water column. And surveys have been done to determine how many fulmar carcasses are washed up on the shorelines around the northern hemisphere that contain plastic. And what you can see here is that around about 90% of the fulmars which have been studied contain plastic items in their stomachs. Sometimes the number of plastic items is quite amazing. Here you can see the record, and these are figures from NOAA, is some 84 pieces in an individual bird. And what the effect of this is to cause gut blockages and gut damage, and potentially the transfer of contaminating chemicals into the tissues of the birds. Again, another very obvious example of the kinds of harms that plastic can do is this image here of entanglement. Entanglement in plastic nets and ropes is estimated to cost the lives of around 1 million seabirds every year and upwards of 100,000 marine mammals, and these are figures from the United Nations. If you consider that there are around 5 million harbour seals across the globe every year, you start to see that these figures are quite important in numbers. And of course, larger pieces of floating debris floating around in the ocean currents can provide novel substrates for invasive species, taking them to areas of the globe where they would not normally reach, and taking microbial communities and different microbial communities and potentially pathogens to those areas as well. But the topic for the rest of my talk is not the macroplastics, but the microplastics that form from those larger pieces of plastic as they become degraded by the actions of wind, tide, and UV light. And by microplastics, and I'm talking there about anything that's smaller than a size of about 5 millimeters, so this takes in plastics that have broken down to the micro scale and to the nano scale. We're talking about microscopic pieces, fragments of things like bags and ropes. We're talking about fibres formed from clothing, from ropes, and from things like smoked cigarette butts. We're also talking about items that have been deliberately synthesized to be of a small size. So things like the scrubbers that we put into our face washes and our shower gels, so-called cosmetic microbeads. All of these things can work their way through the sewage system and find their way out into the oceans. And we know that these micro-scale plastics are increasing. These are figures from Goldstein from 2012, and these were surface trawls taken from the northwest coast of America. And what you can see is that they document the number of microplastics collected from those surface trawls in the 1980s. And what you can see here is a comparison of the number of microplastic particles fished from the same surface trawls a decade later. And you can see that there's been a significant increase in the number of plastic particles present in the ocean surface. And this isn't just an effect that you see off the northwest coast of America, but we see similar effects if we go back to those gyre areas, the great oceanic gyres across the main oceans of the world. We find that where we can see large floating islands of large plastic pieces, if we look, we can find smaller plastic pieces as well. So where you can see plastic, the concentration of microscopic particles of plastic is also likely to be high. And why is this a problem? Well, as you've seen from the previous presentations today, the dimensions of microplastic overlap with the kinds of particulate matter that become important to marine animals and also to biogeochemical processes. So, for example, we can see millimetre-sized pieces overlapping with the size preference for small fish and crustaceans. We move down to the micron on the nanoscale, and we start to see particles that become important for organisms such as filter-feeding bivalves and different planktonic species. So the key question there is that microscopic particles can be ingested, and they have the potential to enter the marine food web. So that is the first question that we set out to address in my research lab. We wanted to understand whether microplastic could be ingested, and if so, by which organisms. And to be able to do that, we studied a range of different species throughout the water column to try and understand what parts of that marine web might be most vulnerable to ingestion. First experiments I'll tell you about are based in marine bivalves. Bivalves can filter huge volumes of water and remove particulate matter, cleaning coastal waters. So if there are particles floating in the water column, they're very likely to be ingested by bivalves. And you can see in these images that we found that particles certainly are ingested. They can pass from the gills of the mussels into the gut. Here you can see particles in the gut, fluorescently labelled. And from the gut, they also pass into the digestive tubules. And here you can see two different sizes of plastic particles that have passed through the gut into the digestive tubules of the mussel. And we also found that they can pass from the digestive tubules and be translocated into the tissues of the animal as well. And you can see here images of plastic particles that have moved into the hemolymph, into the circulatory system of the mussel, and they've been engulfed and ingested by hemocytes, the blood cells of the mussel. And they can remain there for quite some time. When we studied how long those particles might be retained within the blood cells and within the tissues, we found that up to 48 days after an initial feed of microplastic particles, we could still find them present in the tissues of the animal. And at that stage, we stopped the study. So for all we know, they could have been there for longer. Now, this is interesting because it suggests that if they're retained within the tissues of a simple organism, then they could be present and could move up the marine food web. So the next experiments that we conducted sought to determine whether trophic transfer of plastics could be occurring. Could they pass from organisms lower down the food web into others? And the organism we decided to study to look at that was this. This is the common shore crab. This is an omnivorous scavenger on the floor of the ocean, and it frequently feeds on mussels. So what we did was we allowed mussels to ingest plastics from the water, and then we used those mussels and fed them to crabs. And then we traced what happened to the plastic particles that the crabs had ingested along with their mussels. And what you can see from this diagram is up to 14 days later, the tissues of the crabs still contained large numbers of microplastic particles. And we could see that those particles were being actively ingested or egested right up to 14 days after the initial feeding experiment. So they were present in the faecal pellets, they were being expelled actively from the body. You can see some of those particles in these faecal pellets here. And what we then did was we traced back through the body of the crab using bioimaging techniques to determine where they might still be located. And you can see in this image that polystyrene microspheres were still present, wedged within the setae of the gut. We can look at that image in slightly more detail. This is a hyperspectral imaging scan using bioimaging equipment we would normally use for medical purposes to determine where in the body of the crab these microplastic particles were retained. And you can see that they're tangled in with these foregut setae. They've not been absorbed, but they've been embedded, enmeshed into that entangled area. And you can use the same technique to examine the gills of the animals and to examine the other tissues of the animal. And what you can see here is that plastic particles are stuck all over the surface of the gills. So this is up to 14 days after an initial feed of plastic particles in another organism. And here are the plastic particles coating the surface of the gills. And what this implies is that the electrostatic or hydrophobic interactions between the particles and the surface of the gills is strong enough to withstand the effects of the gill rakers. Gill rakers are special modified setae which the crab would usually use to keep its gills clean. Okay, so I think that proves, firstly, that microplastic particles are present in the ocean, and secondly, that they are bioavailable to different marine organisms. But it doesn't necessarily tell us what harm those plastics might be doing. And to do that, the experiments I'll show you next relate to marine worms that are found in the benthos, that are found in the sediments at the ocean bottom. And the reason for our turning our attention there is here, and I'm just going to try and start this video for you. Oh, it's not going to work. There we go. Now this is an image taken with a remote operated vehicle. This is courtesy of a colleague, Rachel Miller, from the Rosalia project in the US. And what she's done here is taken her RUV down to the floor of Newport Harbour. Now Newport Harbour is one of the most prestigious areas in terms of yachting off the coast of America. And what you can see here is if you look at the ocean bottom, there's a vast trash reef of plastic waste. So this extends for several kilometres, and doing some simple modelling of the kinds of concentrations of plastic waste that were found, she's estimated that there are some 41 million plastic items for every square kilometre below the waters of Newport Harbour in America. So the sediment is really somewhere where we need to be determining what might be happening. So to study that, we turn to an organism that lives in the sediments and is found ubiquitously in coastal sediments, and that's the Arenicola marina lugworm. Here you can see it. And what we did was we grew these lugworms in mesocosms in which the sediment had been spiked with plastic. In this case, we used unplasticized PVC. So the PVC particles were checked, or they were chosen to be of a similar size in every other respect to the sediment particles. Studied the animals for up to a month and determined what biological effects might be occurring. And the first of those is that even at only 5% PVC added in with the sediment, we found that there was a significant reduction in the feeding activity of the animals. So there was something about the presence of plastics that the animals did not like, and they started to reduce their feeding. Your obvious conclusion is that, well, they're reducing their feeding because you've reduced the nutritional content of the sediment. But we repeated the experiments, and instead of putting PVC, we put clean quartz sand into the sediments instead. And what we found was that that didn't affect the feeding activity of the animals at all. So it wasn't just that we had reduced or changed the organic content, it was something about the presence of the plastics that the worms didn't like. And that reduction in feeding transferred itself to a reduction in the lipid stores that the animals were able to make. So you're looking at a key prey animal for birds and wading birds and fish, and it's putting on less lipid if there's a small amount of plastic in the sediment. And when we went on to try and study what could be happening, why they would be putting on less weight and feeding less if there was plastic present in the sediment, what we found was that it was taking longer for them to process what was in their gut. So put plastic into the sediment and the lugworm takes longer to feed and ingest its gut contents. So very similar results to the kinds of results that we were getting both with the mussels and with the crabs. And this starts to get really quite interesting because it suggests that organisms with a wide range of different feeding habits can experience similar effects. Okay, well, the last example that I wanted to show you was studying the ecological effects or the ecological impacts in another area of the water column, and that's the water surface. So we started to study the effects of plastics on zooplankton. And what forced these studies was our finding from several of the surface trawls that we'd been conducting ourselves, that the smaller the net you used, the more plastic particles you would find. So we use a 300 plankton Houston net, we find a certain number of plastic particles in the water. Use a smaller net, you'll find more plastic particles. Use an even smaller net, you'll find even more. So the conclusion there is that the number of particles at a very small size, which is within the size range of the prey and the food habit of zooplankton, increases exponentially. And perhaps unsurprisingly, when we started to conduct feeding studies, we found that a wide range of different zooplankton species will readily ingest plastic. You can see in this diagram we've got a bivalve larvae. We've also got a decapod larvae. We've also got more permanent inhabitants of the zooplankton. We've got a copepod here. This is Centropoges, readily ingesting plastic particles. Even when they're offered in association with other food items, they're readily ingested. And what we can start to do then is we can start to determine what factors in the plastic particles makes them likely to be ingested and what the selectivity of some of these organisms might be. What you can see in this image is this is Centropoges, which we've been feeding microscopic beads purified from face wash. So many of you may use this face wash yourself. Feed the microbeads in it to Centropoges typicus, and it really likes to eat these plastic particles in preference to food items. We're also able to start to study things such as size selectivity, and you can see here that different copepod species, some of them show greater size selectivity across different size ranges than others. So Acacia particularly prefers smaller sizes of particles, whereas Tamara is quite happy to eat particles of a wide range of sizes. And we can also see that non-selective species can also ingest plastics. And this is an image from experiments conducted by one of the postdocs in our lab, Matthew Call, and he'll be giving a talk about this subject tomorrow morning, if you care to go and listen to it. And what we can see here is a doliolid. And doliolids, along with salps, are typically seen as some of the vacuum cleaners of the surface of the ocean. So doliolids have a very muscular pharynx, they have a mucus net, and they pull particulate matter into their gut. And what you can see here is that very strong feeding current pulling small microscopic polystyrene particles in through the feeding current, and here they are entering the gut of the animal. And of course, looking at these images, it raised the question for us, well, if they're entering the gut of the animal in such high quantities, clearly they're being digested and they're also being ingested. And when we look at the fecal pellets of doliolids and of other zooplankton species, we find that if there is plastic in their diet, we can see plastic particles embedded in their fecal pellets. And as many of you will be aware, the fecal pellets of zooplankton species and many other species are very important in terms of the ocean, in terms of organic carbon cycling. So pulling that organic matter, that carbon, from the surface of the ocean down into the ocean depths. And the final experiments that we've been conducting that I wanted to show you were starting to determine whether a diet that included microscopic plastic, which clearly typically will have a lower density than the normal carbon food substances of the animal, can change the density and the sinking rate of those fecal pellets. And here we can see these are normal fecal pellets. I think these results are from Calanus, from a small copepod. And these are fecal pellets with a diet in which polystyrene microplastic particles have been added. And you can see that there's a very clear differentiation between the sinking rate and the density of the fecal pellets with plastics and without. And you can also see that the presence of plastic starts to make the fecal pellets become more easily degraded. So they break up. And the two implications of these results are that the organic carbon will no longer be being pulled down to the ocean depths, but will remain floating in the ocean surface. Okay, so that's a bit of a whistle-stop tour through some of our recent research. What I hope I've been able to show you very quickly is that microplastics are present in the ocean, and we can see from the research from our own team and from many others that they are bioavailable to a wide range of marine animals. They can enter the food web through trophic transfer. They cause a wide range of different biological effects, some of which may be predicted and some of which may not be predicted. And that we're still in the infancy of understanding what the ecological consequences of the ingestion and the release into the marine environment of such huge quantities of an anthropogenically derived substrate can cause. So thanks very much for asking me to speak. I'd just like to thank all of the other people whose research has gone towards what I've presented to you today. Thank you.